Hey guys, Professor Davis here, and we are going to be looking at molecular biology today. So with this, we're going to break this into a couple of sections. We're first going to look at the scientists that are responsible for kind of the discovery of DNA structure along with its function. So we're going to discuss them. We're also going to look then at DNA replication, how DNA has the ability to replicate itself, which we talked a little bit about back when we looked at mitosis and meiosis, in the fact that in the S phase, we do see replication take place. We're then going to talk about the central dogma here, where it's called gene expression. We're going to look at transcription and translation, and then we're going to talk a little bit about some mutations. So our little introduction here, it says beginning in the 20th century, there was a race to determine what the genetic material was. There was a debate on whether the genetic material was a protein or if it was a nucleic acid. So they weren't really sure what was happening here. Scientists did know that the genetic material stored information. They did know this at that point. And they knew that it could be passed on from one generation to the next, okay, from parent to offspring. They also knew that it had the ability to replicate. It was able to copy itself prior to cell division, the whole idea of mitosis and meiosis. And they also knew that it underwent rare changes. There were times that mutations did occur, changes happened, and then this is known as microevolution. Small changes were taking place within the DNA. But the thing is, they didn't know it was the actual DNA that was doing this. They weren't sure, again, if it was a protein or a nucleic acid. So when we look here, the first one is from 1869. Mechner was a Swiss chemist, and he was able to be the one who isolated what he called nuclein. Nuclein was the substance from the nuclei, the nucleus of pus cells. He was able to pull that out, and he started looking at it microscopically. And he realized that it was very rich in phosphorus. So there was lots of phosphorus in this nuclein, but no sulfur. Now, nucleic acids and proteins were both found inside the nucleus, but which one had most of the genetic information? So nucleotides, remember, are the building blocks or the monomer of nucleic acids. Whereas, when we look here, amino acids are the building blocks for proteins. The key thing to look at in these two structures is that the nucleotides are the ones who had the phosphorus. They're the ones that had this high level, whereas sometimes amino acids might, but they were the ones in that R group to contain most of the time sulfur. So this was the first time that we saw that most of the chemical makeup that was inside the nucleus was leaning towards the nucleotide side instead of proteins. So nuclein was not a protein, and later it got renamed to a nucleic acid. The next one was a bacteriologist, and his name was Griffith, and he experimented with mice, specifically with also Streptococcus pneumoniae. All right, so he was looking at strep pneumoniae with these mice. Now, strep pneumoniae comes in two kind of strands. The first strand is called the S strand, or the smooth strand, and it gets its name because it has a special capsule around it. This is what makes it more virulent, meaning more dangerous. It makes us sick more with this, and it makes it more invasive. The R, or rough strain, has no capsule, therefore it doesn't make us sick. All right, so the S strain was actually going to be able to kill the mice, whereas the R strand did not. So what he did is, in his first part of the experiment, he took the S strand, that smooth strand, and he injected it into the mouse. Okay, and ultimately, because this S strand is the more virulent strand, it's the more deadly strand, the mouse died. Okay, so S strand was added in to the mouse died. When he took the R strand and he added it to the mouse, okay, he injected it in, the mouse lived. Okay, and so when we look at this, the mouse was able to live, even though it was injected with strep pneumoniae. So he took it a step further and he said, okay, well, what if I kill the S strand first? I'm going to heat it up, I'm going to kill it, and then I'm going to inject it into the mouse. What will happen to the mouse? Well, the mouse lived. So this told us that technically that bacteria was a living thing and we were able to kill it with heat. So he said, all right, how about if I take that dead S strand, okay, I kill off the dead S strand, but I mix it with the R strand. In both cases, the dead S strand and the R strand, the mouse lived. So he's thinking, well, maybe the mouse will live. But the issue was the mouse died. And when they cultured out of the mouse and looked at what was left behind that actually killed it, the strep pneumoniae, he found that there was live S strand in the mouse. This told us that this was able to be where the R strand was able to take in the DNA from the S strand and mutate itself, change. So what happened? Did the S strand become alive or did the R strand mutate? Well, the R strand mutated. So the conclusion is that the genetic material, DNA, can be transformed. It can be changed. Now, further scientific studies concluded the following stuff, that there's two types of nucleic acids. We call that DNA and RNA. The monomer, or the building block of DNA and RNA, is now called the nucleotide. And the basic structure of the nucleotide is a phosphate group connected to a pentose sugar connected to a nitrogenous base. Now remember that pentose sugar can be deoxyribose sugar in DNA, or it can be ribose sugar in RNA. Also remember those nitrogen bases, they a lot of times get represented by a letter. So in DNA you have A, T, G, and C, and in RNA you have A, U, G, and C. So there's two types of nucleotides that are present. You have what we call purines and pyridamines. The purines, guys, are the adenine and the guanine, the A and the G. Those are the purines. The pyridamines are going to be mostly thymine and cytosine. Now, one way to remember this, and I have it highlighted here on the slide, is that the Y's are underlined. All the ones that have Y in it are related. So pyridamines are thymine and cytosine. Now, remember that uracil takes the place of thymine, and that's why it is also in this group. So in DNA, A is always going to pair with T. So a purine is always going to pair with a pyridamine. G is going to pair with C. In RNA, you have the A, you have the G, you have the C, but instead of T, you have U. So there's no thymine in RNA, there's uracil in RNA. So guys, this chart should look familiar. We talked way back about this back in the chemical lectures, the chemical videos, and this chart was in there. So you have DNA and RNA. The type of sugar is what makes them different, as well as their nitrogenous bases. And then, of course, their shape or structure is different. So if we look here, deoxyribose is the sugar in DNA. They are going to have the bases A, G, C, and T. And they do have one phosphate group per nucleotide. Now, when we put these together, do remember that they are going to make a double helix. Okay, double-stranded, meaning it has the two strands, and they're connected in the middle like A to T, G to C through hydrogen bonds. Okay, so hydrogen bonds. Also remember the sides have what we would consider a sugar-phosphate backbone, because you'll notice it goes sugar, phosphate, all the way down. Remember too that the function of DNA is to store the genetic information, and that information is ultimately to help make proteins. On the ribonucleic side, or RNA side, the sugar is ribose. We see that it's going to have the bases of A, G, C, but instead of T, they have U. There is still one phosphate per nucleotide, and it is going to be a single strand. You will still notice, though, that there is a backbone of sugar, phosphate, but then you'll also notice that it's a single strand. Now, the purpose of RNA is to carry out the actual production of proteins. The RNA is required in order to make proteins. So this brings us to another scientist. It's called Chargaff. Chargaff was in the 1940s, and he studied the molecular structure of DNA. He's the one that came up with some rules here. He said if A equals T, then G has to equal C. This is known as complementary base pairing. A purine bonds with a pyridamine. So we always have a purine bonded with a pyridamine. This explains the consistency of DNA in molecules. So we're able to see consistency where A equals T, G equals C. Another thing that he was able to come up with math-wise is A plus G, okay, is going to equal T plus C. So your purines are always going to equal your pyridamines. So in this case, you'd have 50% purines, 50% pyridamines. Each species has its own constant base composition, and this explains what we call the variability found in DNA molecules. So there's consistency, but there's also variability between species. All living things have DNA, but our DNA is very different than other organisms. So this gives it the variability. So if you take a look here, this is looking at Chargaff's DNA database where he was looking at various species. He's showing you how A and T and G and C compare to each other. Remember his rules that A equals T and G equals C, whereas A plus G equals T plus C. So the application here is in Homo sapiens. There is 31% A, there is 19% G, 32% T, and 18% C. This gives you a 50% purines and 50% pyridamines. If an organism's DNA, here's a question for you, a little quiz. If an organism's DNA has 22% adenine, so 22% A, what percentage of thymine will be present? Well, guys, if A equals T, what would T be? 22%. And what percent of guanine would be present? Well, A plus G equals T plus C, so this is 50% where it should equal. It's already got 22%, so what's 50 minus 22? That would be 28. Okay, so you could use his math equations to figure out A, T, G, and C if you are given enough information. Now this brings us to the next set of scientists in 1952 named Hershey and Chase. They were studying bacteriophages. Bacteriophages are viruses that are going to infect bacteria. So bacteria is not even immune to getting sick. These are viruses that have very simple structures. They have a protein coat and a nucleic acid core. That nucleic acid core is made out of DNA or RNA. Bacteriophages infect viruses, so bacteria eventually bursts and releases all these new viruses that they have now made. Now, what they did is they labeled 2 batches of these bacteriophages. They put in one of the groups, they tagged it with radioactive sulfur, which is the protein part, as DNA, remember, does not contain sulfur. On the other group, they tagged it with radioactive phosphorus, which is found in DNA. So then they were allowing the batches to infect cells, and they examined to see which molecules entered the host. Was it the sulfur molecules or was it the phosphorus molecules? And they found that it was phosphorus. And so they knew that the DNA entered into the actual new cells versus the proteins. Proteins did not. And this gave us very conclusive evidence that supported what some had proposed in the past. So guys, this is just giving you a look at how the experiment worked. You can see here that they had the bacteriophages tagged. In the top one, they tagged the DNA at that green. And in the bottom one, they tagged the protein with the red. They have then allowed these viruses to infect cells. And when they looked at the new cells, you will notice that the only thing that got passed on was the green at the top. The actual red protein did not get to enter the cell. Now, this doesn't mean that the cell wasn't infected. It just proved, though, that the DNA is what enters, not the protein. The next one we want to look at is 1953, Rosalind Franklin. She was responsible for kind of coming up with the idea that DNA was a helix. It had a spiral shape, but that it was actually a double helix. There were 2 of them. She used X-ray crystalline technology, where she shot X-rays through crystals and diffused the light in order to get the idea of what DNA looked like. She was an English chemist, and her research implied that there was a helical structure, like a spiral staircase structure. Now, this led to more discoveries by Watson and Crick in 1953. Now, they received the Nobel Prize for determining the molecular structure of DNA in 1962. Now, guys, look what they did. They used Franklin's work of the double helix as well as Chargaff's work with his complementary base pairing, and they came up with this particular structure. Now, in 1969, we were able to actually look at it underneath the microscope, and we found that they were correct. Now, they received the Nobel Prize, but Franklin did not. And mostly the reason being is that she had already died. You cannot receive the Nobel Prize if you are deceased. And so this was part of it. But there was a lot of issues with them potentially stealing that work from her. And so this is kind of what happens sometimes in that scientific community. But Watson and Crick did come up with this double helix. They also were the ones that started talking about how DNA replicates. So with DNA replication, they were looking at this process and they were trying to figure out how it happens. They know that DNA replicates, meaning that it makes more copies of itself. And they knew that this happened during the S stage of interphase. But they weren't exactly sure how this occurred. Now, there were several models that they were looking into, and Milson and Stahl in 1958, they finally did an experiment with E. coli using nitrogen and a heavy isotope that helped prove which one of these actually works. The first one was the conservative model, where they said the old DNA just gets made into new, so the two old are together and the two new are together. On the other end of the spectrum, you had the dispersive, where they just pieces of new were put into pieces of old. In that case, and the case for conservative replication, both of those allow for a lot of mistakes to occur. We actually found that semi-conservative, the middle one, is really how the cells do their replication. They take an old strand, and on that old strand they build a new strand. So there's always something to proofread against and to check their work. So an old strand is always paired with a new. So let's talk about the steps of how this works. Okay, so now as science has progressed, technology has progressed, we now know this is how it works. So steps in DNA replication using the semi-conservative model. Step 1 is we have to unwind the DNA. Remember, it is a double helix spiraling around. We need to unwind it. Okay, this way we can gain access to those nucleotides. So these hydrogen bonds are going to break between the nucleotides. This is done by an enzyme called helicase. Helicase actually looks like it's unzipping the DNA molecule. In step 2, we have our complementary base pairing. We are going to add those bases and pair them up. Enzymes do this as well. This enzyme is called DNA polymerase. We know it's an enzyme because it ends in ase. It's responsible for bringing in the appropriate base pair. So if there's an A over here, it's going to bring in a T to match it. If it's a G, it's going to bring in a C to match it. Okay, and it's always going to do this in the 5 to 3 prime direction. That's what that little mark means, is prime here. So the 5 prime to 3 prime direction. Now, if you'll notice, because of this, the DNA polymerase works in opposite directions, kind of like a 2-lane highway. Okay, where if you're driving between here in Lovington or here in Tatum, which are these small towns, there may be like a 2-way road where cars are going north on one way, south on the other. Okay, and so they're going opposite directions. The same thing here on the DNA polymerase. On the top picture, you'll notice it's adding a nucleotide at a time. On the bottom one, it's going to add it in pieces. Those pieces are known as Okazaki fragments, and they are named after the scientists who first saw them. Right, and so they do the same process of complementary base pairing, they just do it a little bit differently. The third step is we're going to join the old strand to the new strand. The enzyme again responsible for this is DNA polymerase, and they are going to form those hydrogen bonds between the old strand and the new strand. Last but not least, they are going to proofread their work. So an enzyme, DNA polymerase, is going to check for any errors or problems. In other words, it's going to look to make sure there's no wrong pairings. Remember, A always should pair with T and G should always pair with C. But if something is wrong, we want to fix it. Okay, we want to correct the problem. And this is where the proofreading comes into play. And so most of the time we're going to correct our mistakes if they are present. So here, DNA replication is enzyme driven. Okay, so enzymes run this. Ergo, there's high fidelity, meaning it's very fast and it's very accurate. Base mismatching only occurs about 1 in every 100,000 base pairs. And after proofreading, it only happens in 1 in a billion. Okay, 1 in a billion. Errors that are not caught, we call those mutations. So if the errors are not caught, we eventually call those mutations. Now guys, some mutations are caused by environmental mutagens, meaning like your environment helps mutate your DNA in your cells. And some are just spontaneous, meaning they just happen and it's nothing we could have done to stop it. Mutations that can either happen in the somatic cells. If they happen in the somatic cells, you do not pass those on from parent to offspring. But if they do happen in the germ cells, the egg and sperm, they do get passed on to the offspring. So somatic cell mutations can lead to development of what we call cancer, but germ cell mutations can be passed on to subsequent generations, so future generations. So most mutations, guys, are detrimental or harmful. However, some mutations could be beneficial. This is what allows us to have a lot of differences within the human population. And this is what natural selection and evolution looks like. So some concepts when we're talking about DNA replication. First of all, there's what we call the telomeres. Telomeres are at the end of DNA, and they do not code for proteins. So telomeres are at the very end of the DNA, kind of like a cap on the end, and they do not code. Now, each time the DNA gets replicated, and that occurs on a regular basis, the telomere region gets shorter and shorter. Mammalian cells like us can divide about 50 times before they undergo apoptosis. We talked about this as being like cell suicide due to the loss of their telomere. Once the telomere is all gone, we would then start losing important genetic information, and so this just stops the process of division. Cancer cells, however, can produce their own telomeres. This is what makes them have their abnormal growth, and they can keep doing the cell cycle over and over and over again. Now, this is something worth studying, and we don't know exactly how it works yet, but this is how cancer cells are able to bypass or override a lot of those checkpoints that we talked back about in the mitosis chapter. All right, so this brings us to gene expression. This is where the genotype affects the phenotype. Remember, your genetics, those letters affect what we see, the physical appearance. Now, the function of your genes are to code for a protein. Remember that this is going to be where they make the amino acid chain. That chain folds into a polypeptide and then ultimately into a protein. We talked about that back in the chemistry chapter as well. So again, the genotype is the gene. It ultimately is going to affect the phenotype, which is the protein. Now, guys, DNA codes for specific sequences of amino acids, and this is what helps build those polypeptides and ultimately those proteins. So a gene is being expressed when the cell product is being made via transcription and translation. So down here at the bottom, it shows you that DNA is going to be copied into RNA. This process is known as transcription. And then RNA to protein is known as translation. Now, guys, if you'll notice, DNA and RNA are the same type of molecule. They are both nucleotides. They have some slight differences, but we're transcribing the information. We're copying it down. We're not going through the process of translating it yet. We just copied down the code. Now, when we go from RNA to protein, we have to translate it into a different language. This would be like translating English into Spanish or English into Swahili or whatever it might be. You are translating it into a different language so somebody else can understand. Guys, your cells don't understand DNA and RNA or nucleotides, but they do understand proteins. So we're going to translate it from RNA to a protein here. Now, guys, this is known as the central dogma in biology. Gene expression is not this simple like I'm going to show you, but it helps to explain it in order to kind of see the basic process. So DNA is to serve as a template, okay, like the guide, the copy of instructions, where DNA replication and protein synthesis. You can see in this picture, we see DNA to DNA. That's replication. But if we go from DNA to RNA, RNA to a protein, that's going to be transcription and translation. Now, not only does DNA replicate itself prior to cell division, it also is going to control all cellular activity. So let's look at our summary of gene expression. We're going to start with DNA. DNA is going to get copied into RNA. This process is known as transcription, and transcription takes place inside of the nucleus. So DNA to RNA, transcription, it happens inside the nucleus. Now this RNA, because it is a single strand, can now leave the nucleus. When it leaves the nucleus, this RNA strand is going to come in contact with a ribosome, and translation is going to occur. We're going to read the code and translate it into amino acids. And you can see that those amino acids make up that primary structure of a protein that looks like the string of pearls. So let's talk quickly about transcription first. Now, transcription does take place inside of the nucleus. It's going to use an RNA transcript, and we're going to make it from DNA. So the RNA in red is going to be made from one side of the DNA in blue. This is also enzyme driven, just like DNA replication was. It's going to be RNA polymerase here. RNA telling you we're making RNA. Now, on the DNA, there's going to be a region that is called the promoter. This is the region of the DNA where transcription starts. The copying of all those nucleotides will continue until a terminator is reached. This is the region of the DNA where transcription stops or ends. So guys, if my DNA has T, C, G, and A, the complementary RNA strand is going to be A, so T pairs with A, C will pair with G, G will pair with C, and A will pair with U because there is no thymine in RNA. So A is going to pair with U. Now, there is a process that happens after we first make this first messenger RNA or mRNA. There's going to be an editing phase where we're going to take a part of this and we're going to cut pieces out. Those pieces that get cut out are called the introns. They get clipped out and they stay in the nucleus. They do not get to leave. The exons then get spliced or pushed together, and this is going to be what gets to leave the nucleus. So the exons get to exit the nucleus. They're the ones being expressed. This is also enzyme driven, and the enzyme is called a spliceosome. So now let's talk about translation. This takes place in the cytoplasm. This is where RNA is converted into a polypeptide protein. There are 3 types of RNA that are going to be be important in this process. The first is the red ribbon, which we call the messenger RNA. This contains the codons or the code, and they are in 3 nucleotide sequences. So like CCA, GGU, they're going to be in 3s. There's also going to be transfer RNA, and these are kind of shown in the turquoise color. They contain the anticodon, the opposite of the codon, and they're going to be the ones who carry the little purple structures, which are the amino acids. Now, where are they going to carry them to? They are going to transfer or carry them to the ribosomal RNA, the ribosome. This is going to be where they match the codon to the anticodon, and we actually see the building of the new protein. So if my codon in messenger RNA is U, C, G, A, in tRNA, you're going to see that the U pairs with A, C pairs with G, G pairs with C, and A still pairs with U because this is still RNA. Okay, so this is still RNA. So this process goes first with chain initiation, where we start to build the actual protein, and this starts with the codon AUG. AUG is known as the start codon. The polypeptide chain just elongates in step 2 and gets bigger and bigger, longer and longer until we see chain termination. This is where the codon for the stop signal has been reached, and we will stop making that protein. So guys, there's a lot of terms that come into play here, so let's review some of them. The making of another DNA molecule from a parental strand is known as replication. Making RNA from a portion of DNA segment or a gene is known as transcription. But building of the polypeptide molecule from RNA is known as translation. So these are your 3 main processes. Remember that DNA contains the unit of heredity that we call genes. mRNA is going to carry the codons. Messenger RNA is what that M stands for. The tRNA contains anticodons and carries a specific amino acid, and the T stands for transfer RNA. And then this RNA matches the anticodon to the codon, and this is our RNA or ribosomal RNA. All 3 of these RNAs must be present in order to build a protein. And amino acids, remember, are the building block of that protein. Now, a codon, guys, consists of 3 nucleotide bases, and it's known as like a triplet code. But the anticodon consists of 3 nucleotide bases that are complementary to the codon. Anti means opposite, so they're complementary. A with U, G with C. So why does the codon consist of 3 nucleotide bases? Well, guys, RNA contains 4 different nucleotides, and there are over 20 different amino acids. So we can't just have a 4 code. If they were all 1 nucleotide long, they would only have 4 codes. So if 1 nucleotide was there, 1 amino acid, we'd only have 4 codes. If there were 2 nucleotides, we'd only have 16 codes. And again, 4 times 4 is 16. That's still not enough because there's 20 different amino acids. So we need to take it a step further and look at 4 cubed. That's 64 codes. This is the one that they utilize. The pairs of 3 help us get those 64 different codes. Now, I do not expect you to memorize all these codes and codons and all. I do want you to learn how to use the chart. So using the chart, you should be able to find the 3 codons on here in order to find which amino acid it codes for. So in this case, let's just say that our codon was GUA. So I'm going to find G, then I'm going to find U, and then A, and that's going to code for the amino acid Val. Now, genetic information is encoded on the messenger RNA, the mRNA, in these 3-letter units that we call codons. Okay, codons. Now, AUG is always the start codon. It's shown in green. So AUG is methionine. It's the start codon. Whereas there are also 3 stop codons, and they're shown in red. So UAA, UGA, and UAG. Those are all going to be considered stop codons, and they stop the process of making the protein. So guys, there's a couple of things that this means the genetic code is. Okay, so the genetic code is what we call degenerate or redundant. More than one codon codes for an amino acid. So I have 61 codes that code for 20 different amino acids. So GGU, GGC, GGA, and GGG all code for one amino acid known as glycine. Okay, and so this allows it to be redundant. This also allows for what we call the wobble effect. The third base is less discriminatory for the amino acid than the other 2. So this third position, whether it's the U, C, A, or G in this box, it allows for the wobble effect. If something mutated that last letter, it could still potentially be a glycine molecule that we're coding for. The second thing that we see with DNA is it's what we consider unambiguous, meaning that each codon codes for a specific amino acid. So example here, AUG always codes for methionine. That's all it does. It cannot code for anything else. So CCU only codes for proline. Okay, whereas also when we look, CAU is going to be histidine. So it just depends on which one it codes for, but it always is going to code for that specific amino acid. We also see that it contains codes for 20 different amino acids. It contains 1 start signal and 3 stop signals. The start signal is AUG, so the first amino acid is always methionine. The stop signals are UAA, UAG, and UGA, and they do not code for an amino acid. Instead, they tell these molecules to stop. Some genetic code is used by virtually all living things. Mitochondrial DNA has a few differences, and this allows for genetic manipulations where we can take genes from one organism and we can put it in another. So we insert it from one organism into the genome of the other to be expressed. An example here is bioluminescence. We can take the bioluminescence gene from jellyfish. This is the gene that makes jellyfish glow, and we can put it inside of a mouse and it can cause the mice to glow. All right, and so we can potentially do this. Now, should we do this? That's a different question. And this is actually what you're going to be asked to do when you look at your bioethical topics, is you're going to tell me what the bioethical topic is, what are the pros, and what are the risks. What are the good things that come out of it, but what are the risks that come out of it? And this is part of that process. So let's look at transcription and translation a little closer. So transcription is a process where we're making RNA from a DNA template. The portion of DNA gets unzipped and the gene is going to be exposed that we want to express. The site of DNA transcribed is called the template, and the site of DNA not being transcribed is called the non-coding strand. RNA polymerase then binds to the DNA at what we call the promoter region, where we promote the copying. Complementary base pairing then begins. This continues until it reaches a terminator. Once the terminator is reached, we are going to stop copying the DNA. Okay, so once it's reached, we're going to stop copying the DNA. Now, the editing of messenger RNA occurs in some multicellular organisms like ourselves. Primarily, mRNA is edited in the nucleus. Matured mRNA goes to the cytoplasm. Enzymes are going to do this, and they are called spliceosomes. Spliceosomes cut out the introns. Okay, they get rid of them, and they rejoin the exons. Okay, so guys, this is actually how they used to edit old camera film for old movies. They would cut the film and we'd cut the scene out and then paste the two sides back together. And this is kind of what's happening here. Matured messenger RNA then gets a cap put on and a poly A tail, and it's ready to then leave the nucleus. The matured messenger RNA then leaves the nucleus into the cytoplasm, and it does it through those little nuclear pores. So guys, a couple important notes about transcription. mRNA is going to be the transcript. It does not become part of the DNA molecule. Okay, so even though we're copying it down, it does not stay attached to the DNA molecule. More than one messenger RNA molecule can be made from that DNA template. I can end up making 5 copies instead of just 1 if I need to. rRNA and tRNA are also made via transcription. They are just going to get processed in the nucleolus before they leave the nucleus. So now let's talk about translation. Translation is the process that builds a polypeptide from the messenger RNA. The ribosome, the rRNA, is going to be made of 2 subunits. You have a small subunit and a big subunit, and they come together. They sandwich the messenger RNA. When they do this, this creates 3 sites for attachment. The first is the A site, and this is where tRNA begins to bind when it brings its amino acid. We then have the P site, and the P site is where the polypeptide chain elongates. It's getting longer and longer. And then the E site is where the tRNA is going to then exit and go pick up another amino acid. Okay, so it dropped it off. It's going to go pick up another one. Now, this is something that brings us to the steps. How do we know we need to start the process of making a protein? Well, first of all, we have to have initiation of the amino acid chain. So rRNA attaches to mRNA and advances 1 nucleotide at a time, searching for the start codon and knowing that the start codon is AUG. Once it finds AUG, we start the process of elongation of the amino acid chain. So once the start codon is found, the RNA reads the 3 nucleotide bases called codons, and the tRNA brings in the appropriate amino acid by the codon. This then brings us to termination of the amino acid chain. rRNA reads the stop codon. Once it reads the stop codon, then the mRNA and polypeptide chain is cleaved off. Okay, means breaks apart. So we have all of our parts. You can see here that we have the polypeptide. You have the small and large subunits of the ribosomes that have broken apart, and you have the messenger RNA. Okay, and so this is what you see with termination. So some important notes about translation. More than one polypeptide chain can be made off the mRNA template, and more than one ribosome or RNA may attach to the mRNA at one time. So you can see that here in the picture where the mRNA is running like a string, and it has lots of ribosomes attached, each one making the protein that this mRNA codes for. All right, so if we go back over here, guys, you have DNA replicating itself to DNA. You then have transcription, which is the process of making a template, okay, in the sense of taking that DNA template and making the code for messenger RNA. We then see translation is the process by which that RNA has been used to produce a polypeptide or protein. Now remember that transcription does this through a promoter. This is where the transcription starts, all the way to a terminator, which is where it stops. In translation, you're going to see that this edited phase of this transcript from transcription is going to have the introns clipped out and the exons joined together, so that that messenger RNA is going to come over here and go through translation. Remember that transcription is enzyme driven. RNA polymerase as well as spliceosomes play a role. In the translation side, we see that you have the initiation, which is the finding of the start codon, which is AUG. Chain elongation, where the chain gets longer and longer, until finally chain termination happens, where it stops copying down or making the protein. So the key thing here, concept, is that DNA does control cellular activity, and it does it by dictating the products that need to be made, the types of proteins that need to be made. All right, so guys, this is going to be similar to a lab you're going to be doing where you have your transcription and you have your translation. You'll notice you have your DNA template at the top, you have your messenger RNA strand, and then you also have your tRNAs. These are going to in turn create a polypeptide. So you need to be able to use the chart. So as if we take a look at this, if my messenger RNA says AUG for number 1, what would that actually be if we were to go back to the DNA template? What does A pair with in DNA? Well, A pairs with T. What does U pair with? U pairs with A, and G pairs with C. So number 1 would be TAC. If you go down here and you need to find for number 2, you would use GAG. And remember, this is going to be RNA, so there's not going to be any T's. And so G is going to pair with C and A is going to pair with U. All right, so you need to make sure you remember your base pairing rules. Down here in the number 4s where it says 4A, 4B, 4C, that part is going to be looking at the actual chart. Okay, so you're going to use the messenger RNA, not the transfer RNA, the messenger RNA, and you're going to look up at AUG over here in the chart. So you have A, you have U, and then you have G. That's going to be met. So 4A is going to be met. Okay. If you look at the next one, it's CCC. So if you find where there's CCC, 4B would be pro. Okay, so that's how you utilize this kind of structure. Now, remember that the lines that are happening between these pearls are going to be peptide bonds, because peptide bonds are going to be what we see with proteins, and AUG is going to be the start of transcription. No, is going to be the start of translation. All right, so this brings us lastly to genetic mutations, when there's a change in the base sequencing of a gene. So there's some different types of mutations we're going to look at. The first one's known as a point mutation. This is a change in a single DNA nucleotide. All right, so if the DNA sequence is ATG, what would our codon be? Remember, we're copying it down. So A would pair with U, T with A, and G with C. So this would be UAC. So now I'm going to go to the chart and I'm going to find UAC in the chart, and that is tyrosine. Okay, so that is tyrosine. So I have the actual sequence. I'm going to make it into a codon, and then that codon is going to help me find the amino acid. Now, a silent point mutation is where the mutation might be something like ATA. So instead of being ATG, it is ATA. So the codon would be UAU. So if we go over here, what does UAU code for? Well, it still codes for tyrosine. So this is why it's considered a silent mutation, because the mutation didn't change anything. Okay, so result of a silent point mutation, normal proteins are still provided, and this is because of that wobble effect we talked about earlier. A nonsense point mutation is a little different. So here I had ATC. This is going to be coded as UAG. UAG on the chart is going to code for stop. So this might be where it stops the formation of the protein too early. It's a nonsense mutation. So the results of nonsense point mutations are incomplete or non-functional proteins. The next one's known as a missense point mutation. This is a mutation that we might see where it changed to GTG. So the code is going to be CAC. If I go over here and look up CAC, that is histidine. Now I'm coding for something different. I was coding for tyrosine originally. Now I'm coding for histidine. This result of a nonsense point mutation leads to faulty, dysfunctional proteins. They cannot make their proteins properly. Okay, and so we have to keep a close eye for that kind of thing. Another type of mutation that could take place is called a frameshift mutation. This is where there's an insertion or a deletion that's taken place in just one of the nucleotides. So I know that these aren't all the letters that we use for the periodic table, but remember that they come in codes of 3. So the cat ate the rat. That is a sentence that actually we can know what's going on. We actually can figure it out based on this complete sentence. It makes sense. However, if there's an insertion or a deletion, let's just say if the letter C was deleted. So instead of saying the cat ate the rat, it's the atta tit her at. That doesn't make any sense because it's causing there to be a shift in that 3 pyramid type of sequence, that 3 codon sequence. So here's an example where it has before a mutation. This is what the normal one would look like. Here's what happens if an insertion takes place versus if there is a deletion that takes place. Cystic fibrosis, which we talked about last unit, as well as PKU, those are both involved in what we call frameshift mutations. So questions here. In normal hemoglobin, glutamic acid is the 6th position on the beta polypeptide chain. Okay, so we see it there. In sickle cell, though, valine is the 6th amino acid position. What kind of point mutation would occur in sickle cell? Was it a silent, a nonsense, a missense, or a frameshift? Well, in this case, it's a missense because it still codes for something, but it codes for the wrong thing. Okay, so it would be considered a missense mutation. All right, last but not least, this brings us to a triple repeat expansion. This is another change in base sequencing that can take place. This is where 3 bases get repeated over and over again. So instead of just having that code CAG once, the patient may have where it says CAG, 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 CAG over and over again in their DNA. This ultimately can disrupt protein synthesis and cause issues. So when we look here, the more repeats there are, the more pronounced the signs and symptoms of the disorder are. They only have 2 repeats, it's not as big of a deal versus if they have 12 repeats. The more repeats, the more severe it is. Some examples of these repeated sets are going to be Fragile X, or what they call the CGG repeat. So Fragile X syndrome. Huntington's disease is a CAG repeat, where it repeats it over and over and over again. And then myotonic dystrophy, or CTG, is also going to be a type of mutation. This finishes up molecular biology. If you have any questions or concerns, please let me know.